is that everything that we're talking about after this is all going to be applicable to the things we've covered previously. We're going to be taking what we know about metabolism, taking what we know about microbial growth, and combining them to talk about microbial diversity in a little bit. When we get to microbial control and how all of that works, we're going to be taking our understanding of these concepts and applying them to how do we kill organisms. On your exam, there was a question that said, name 2 ways in which you use microorganisms or their products in your everyday life. I got a lot of answers that said pasteurization. What is the process of pasteurization?
Heating something.
Heating something. What are you doing to it? What are you doing to the bacteria that are in there? Killing them. Do you think killing them is using them or their products?
No.
No. Okay? So if you described a method that was directly killing bacteria, that is not using them or their products in any way. Okay? I got a lot of, I heat my food, I use pasteurization, I drink this product, I use Lysol. Lysol is killing them, that's not using them or their products. So you really want to make sure that you're applying the principles we're talking about in class to these different concepts that are coming up. All right? So let's pick up with fermentation. We covered aerobic and anaerobic respiration. Remember, they had 4 parts: glycolysis, the transition step, the TCA cycle or the Krebs cycle, and the electron transport chain. Both of those processes have respiration in the title. Respiration means they use an electron transport chain. Fermentation is different from this process. Organisms choose to go through fermentation or have no choice and must complete fermentation for 2 reasons. Either they cannot go through electron transport, so in other words, respiration isn't possible, or they do not, typically because they do not have a suitable electron acceptor to complete anaerobic respiration. Some reason they can't complete that process, they go down fermentation instead. So an organism could, in theory, do aerobic respiration to start. If they do aerobic respiration and they run out of oxygen, they don't sit there and go, okay, whoops, I'm no longer completing catabolism, I'm no longer making ATP. They say, okay, how else can I make ATP? I don't have oxygen available. If they choose to go down fermentation instead, that then still allows them to make ATP even though they don't have oxygen present. They would more likely choose fermentation over anaerobic respiration because it's coming back to the final electron acceptors. So being able to walk you through why an organism would complete one of these processes versus another is really dependent upon your understanding of the process. What are we doing? How is that unique? What are the differences between them? Okay? An organism that completes aerobic respiration is not likely to switch to anaerobic respiration because it's 2 totally different final electron acceptors in the end, and typically it's ones that are not used. Okay, so fermentation becomes a very common alternative. Some organisms only go down fermentation pathways. Other organisms go down fermentation pathways when there's no oxygen present or when they can't complete that electron transport or they're missing a final electron acceptor. Okay? Fermentation starts off by completing glycolysis. At the end of glycolysis, you are left with 2 molecules of pyruvate. When you go through, quote, fermentation can take one of 2 pathways. The first pathway is an acidic pathway. And the end product is going to be lactate, which is also called lactic acid. So you're converting the pyruvate into lactate. If we started with 1 glucose molecule, how many molecules of lactate do we make? 2. Okay, because we had 2 molecules of pyruvate we're starting with. The alternative is to go down an alcoholic pathway, where we convert pyruvate ultimately through a process, biochemical pathway, resulting in alcohol as your byproduct. Okay? Note, these processes generate a small amount. You're using a small amount of NADH to go back to that NAD molecule, which goes back to glycolysis and gets recycled inside the cell. All this is a biochemical pathway. That cell could decide it doesn't want lactic acid. It's going to take that and convert it into something else. It could decide it doesn't quite need ethanol. It needs a different alcohol instead. It's all about what chemical pathway the organism has available, what enzymes it has available as well. Here's another way of looking at it. This one's from a different textbook. You see glycolysis. We convert glucose into 2 molecules of pyruvic acid, which is also pyruvate. Organisms will go through fermentation. They're doing this because there's no oxygen present, or they're not able to utilize it. They can go down an acid pathway, known as lactic acid fermentation, and they're producing lactic acid. Or they could go down an alcoholic fermentation pathway and give off alcohol as a byproduct. Most yeast typically drive down the alcohol pathway. There are plenty of bacteria that go down the acidic fermentation pathway. We utilize these guys in making things like yogurt, dairy products. If you are eating dairy, there is lactate in dairy. The reason it's there is because the organisms that were involved in making that product, produced the lactic acid. Yeast are used. You make bread, okay? The reason your bread rises is because those yeast give off carbon dioxide. All those little bubbles of carbon dioxide allow your bread to be really nice and soft and rise and have all those little holes in there. Same thing happens if you're making beer, okay? It's got bubbles because it's naturally fermented and it's producing CO2. You've ever made homemade kombucha or kimchi, okay? Forms of sauerkraut. You're driving fermentation. You typically are driving one of these processes. The more common way to do this is to end up driving alcohol fermentation, okay? They give off gas as a byproduct. The reason they do that is because they're getting rid of CO2. Remember, pyruvic acid has how many carbons?
3.
Oh, 3. And we're releasing it and giving some of it off, okay? So fermentation is a process organisms can use when they don't have oxygen available. Our muscle does this. Ever heard of runners getting cramps? Long distance runners getting cramps? They get cramps because they are running and we do aerobic respiration. Hopefully you realize that. We do aerobic respiration. If you run out of oxygen in your muscle, those tissues don't just go, whoops, we're not doing anything anymore. They go through fermentation instead. You build up lactic acid, and then it results in a cramp due to all of that acid buildup in your muscle. Okay? It's the same process. Fermentation is used to make all kinds of different products. What you will notice here is this, so we're taking that pyruvate. Ultimately, the product that you produce is dependent on what that organism uses as its terminal electron acceptor. All this is a giant game of hot potato and passing around the electrons. Wherever it stops, that final electron acceptor is helping to dictate what products we are producing. Different organisms, here you are looking at the genus. Those organisms are specifically going to drive fermentation down one particular pathway. Okay? Clostridium species, for example. They can be really nasty. C. diff, okay? All kinds of nasty things. But we can use these guys because they complete fermentation. Isopropyl alcohol, they give off rubbing alcohol. You can use them to make acetone, okay? It's all about driving fermentation in that organism. Propionibacterium. These organisms are commonly involved in cheese making. Now, it's the genus. So the different species of Propionibacterium give you different flavor profiles and make different types of cheese. But they do fermentation and they release propionic acid, acetic acid. Some of them give off CO2 and they give you different types of cheese, okay? They are completing fermentation. The organism doesn't care that we use them to make cheese. Doesn't matter to it. All it's worried about is, I need an energy source. What can I break down to make ATP? And I'm using fermentation to do that. The byproducts are things that we then use, okay? Or do something with, or they give off and we just let them sit there, okay? We making sense? We've basically covered the biggest component of catabolism. Breakdown of sugars to release ATP. The easiest thing for an organism to break down, to make ATP out of, is sugar. Sugar is the preferred pathway. Today we set up those tubes in exercise 7. You set up 4 glucose tubes. Guess what? Every one of those 4 tubes is going to turn yellow because every organism is going to break down through fermentation glucose. Glucose is the simplest sugar to break down. So this is the preferred pathway here. If sugar is not available, an organism does not up and die, it breaks down something else instead. So it can break down amino acids, which, remember, make proteins. If you break down amino acids, you can get some amount of compounds that feed into these different components. Today you set up the tryptone tubes, okay? The tryptone tubes, we said, contained tryptophan. If an organism makes the enzyme tryptophan ACE, it breaks down the tryptophan. Breaking down that amino acid is going to give that organism some level of byproduct to then drive through the process of catabolism. If an organism doesn't have the ability to break down amino acids, they can actually break down individual nucleotides as well. It doesn't just roll over and die. It says, what else do I have? This is like you run out of cash and you're searching every nook and cranny of your bag, purse, house, pockets for spare change, okay? You don't just go, whoops, I'm out of money. You're trying to find where can I, get it from, all right? Same thing happens with lipids and fats. They can be broken down to enter into this cycle. We talked about this the very first time we brought up metabolism. We said, look, when you go on a diet, what's the goal? Your calories out has to be more than the calories you bring in. That is all coming back to this process. The food you are eating constantly is being used for carbon sources and energy sources. You're breaking it down to make ATP. If you break down all of the stuff that you bring in, but you're still expending energy, you've got to get the rest of the energy from somewhere. You've got to get that ATP back. You are trying to drive your body into breaking down fat to make ATP to help you lose weight. That's the whole goal. Calories out has to be more than the calories you take in. The whole point of that is that you're driving breakdown of other compounds for ATP production to use those instead. Cells can do the exact same thing. The whole point of catabolism is we have to get ATP. The 4 main types of macromolecules inside a cell are at the top: nucleotides, amino acids, carbohydrates, and fats. The preferred method of breaking down to make the most ATP is to start with sugar, and specifically to start with glucose. When glucose isn't available, these organisms, depending on their enzymes, can utilize other sugars, like we gave the lactose broth today, okay? They can also break down other compounds. It all comes back to what enzymes it has. Can it do electron transport or respiration? All of those play into which pathway it chooses, okay? What questions do we have?
Can I break down the steroids? Like cortisol? Isn't that a lipid technically?
It's ultimately a lipid,
Yeah.
so yes, you can. Yeah. If you've got the right enzymes to allow for breakdown, yes. Not a preferred pathway, but they can absolutely do it. They do, depending on the organism. This is another way of looking at the same exact concept. It's just a more involved diagram. Breaking down of other compounds to feed into this process in some way, shape, or form allows an organism to make some amount of ATP that it can live off of. Okay? If it starts with glucose and it goes all the way through aerobic respiration, that's the most efficient. But you know what? If glucose isn't available and it breaks down more complex sugars into simple sugars that then feed into this process, it still makes a good amount of ATP. It's still going to be able to survive. Today, again, 7.1, you had glucose tubes, you had lactose tubes. Glucose is the more standard sugar. Lactose is a disaccharide. It's two sugar molecules put together. If an organism has an enzyme, lactase, to break that lactose apart into a simple sugar, the simple sugar can feed into this process. Okay? We also used a starch plate today. Really big carbohydrate. Doesn't mean the organism can't do it. It means, do we have an enzyme that allows us to break it down into a smaller sugar that can come on in and be utilized inside the cell? Okay? It's all about making ATP. And now we're going to talk about photosynthesis. I know, right? Photosynthesis in many ways seems like the opposite of respiration. In respiration, we talked about starting with glucose and breaking it down, giving off CO2 as a byproduct, utilizing oxygen as a final electron acceptor. Look at this. Here we're bringing in carbon dioxide, we're utilizing water and sunlight, and now we're producing that molecule of glucose. We're giving off water and we're giving off oxygen. So in some ways it's opposite, but a lot of the pathways are going to look familiar. This is still about electron transport. This is still about making ATP. This is, in this case, about using the energy from the sun to generate ATP production and then generate compounds for the cell. So the whole process of photosynthesis includes two main types of reactions. What are known as the light-dependent reactions and the light-independent reactions. The whole point of the light reactions, this kind of accomplishes both catabolism and anabolism together. So the light-dependent reactions are going to produce the ATP. They're taking in the sunlight and they're generating that into ATP. They are also going to generate reducing power. And in this case, we're talking about a molecule of NADPH. So the light-dependent reactions produce our products that we need for the cell, ATP and our reducing power NADPH. The light-independent reactions are sometimes referred to as the dark reactions or the Kelvin cycle. All means the same thing. The whole point of the light-independent reactions are to take that ATP and the NADPH and convert it into making organic compounds. In this case, think glucose. Okay? So two sets of reactions to accomplish this process. Yeah?
Will we have to know the actual equation? Or no?
You should know the equation. Yeah. You should know what glucose is. You should know what the different compounds are. You should be able to walk through this diagram, okay? This diagram is summarizing the light-dependent reactions of photosynthesis. So what happens here is that you have what are known as photosystems. Photosystems are a grouping of pigments, okay? If this were a plant, you would just think chlorophyll molecules, okay? Bacteria that do photosynthesis can have all kinds of different pigments. They can have green pigments and red pigments and orange pigments. Doesn't matter what color it is, the pigment molecules are going to be important because they are going to be what you're going to use for electron transport. Bouncing around electrons, helping to grab the energy, okay? So yes, in this case, photosystem 2 is listed before photosystem 1. It is not that photosystem 2 necessarily occurs before photosystem 1. In theory, these are all actually happening at the same exact time. But photosystem 1 and the pathway to make NADPH was actually discovered first. And then they realized, wait a minute, this earlier photosystem has to happen before this and involves the breakdown of water through this process, okay? So what happens is water gets broken down. You give off H plus. Water, H. What's H plus?
Hydrogen.
Hydrogen what?
Ion.
Ion, which means it just has what molecule? Or what
Proton.
part? It's just a proton. Okay, so this is 4 protons, and it's giving off oxygen. Okay? Breaks down water, yields protons and oxygen. I think this sometimes helps if you think about plants. Okay? Plants need water. Why do they need water? The water is responsible for the breakdown. Without the water, this process doesn't work. I don't care how much sunlight you give a plant. If it doesn't have water, it's never going to grow. Okay? It will die. So this process yields protons and oxygen. Also think, oh yeah, plants are good because they're giving off oxygen. Okay? They're giving off oxygen because of this process. In this process, when that happens, you have an electron that comes out. And the electron sits in this photosystem at what's known as the reaction center. When sunlight comes and it hits on these photosystems, the energy from the sun bounces through these different pigment molecules. And when it hits the reaction center, it spits out the electron up to what's known as an excited chlorophyll. Okay? Becomes excited. Just a game of hot potato. We're passing around the electron. Okay? The electron goes through a series, in this case, of proton pumps that create a proton gradient. So look at this. The proton pump creates lots of proton on the outside. The proton is actually going to flow back in through an enzyme, and it's going to make ATP in the end. So it says proton gradient formed for ATP synthesis. So after photosystem 2, we're making ATP through that proton pump. The electron allows that process to happen and then goes to another excited reaction center. It sits and waits till the radiant energy sits down and bounces all around. When it hits this, the electron pops out again. Now, it's going down through a series of another bunch of electron carriers. And in this case, it forms the reducing power NADPH by sending it through an enzyme known as NADP reductase. The two products of the light-dependent reactions are ATP and NADPH. The ATP is produced after photosystem 2. The NADPH is produced after photosystem 1. We said reducing power. In respiration, we talked about NAD and FAD. Here we are using NADP. P, think photosynthesis. NAD and FAD are used in respiration. NADP is used in photosynthesis. So on the exam, if it says which carrier molecule, what reducing power are we generating in photosynthesis, you better have that P listed there, okay? The P stands for photosynthesis. This carrier is our little taxi, our little Uber. I know you made fun of me for that one last week, okay? Used in photosynthesis specifically, okay? The other set of reactions are called the dark reactions. What does this look like to you? Looks like the Krebs cycle. All this is a cyclical biochemical pathway. Remember the Krebs cycle? We were giving off carbon dioxide. This is opposite. Here, we're putting in carbon dioxide, and we're going to build cellular components. We're going to make glucose, we're going to make other sugars, we're going to allow that cell to grow. If you're talking about a plant, you're going to make things like cellulose. You're going to make compounds that allow that plant to grow. This particular pathway adds on 1 carbon atom each time it turns. So if it is going to add on 6 carbon atoms, think glucose, C6H12O6, okay? It's got to turn 6 times in order to allow that process to occur, okay? So your molecules here are all listed for all 6 that have to come in to make 1 glucose molecule. Look at what you're breaking down. ATP is going to ADP. NADPH is going to NADP. We're breaking down more ATP over here. All of the components of the light-dependent reactions are being utilized in the light-independent reactions. So you've got to make the ATP first. You've got to make your NADPH first. And then you can utilize them over here to make sugar. Okay? Organisms, bacteria that do photosynthesis, drive this process. Some of those can also do respiration and other things, okay? But they're getting the energy from the sun. They can get their energy from the sun, okay? As that energy source. What do we think of photosynthesis? You can find 1 million videos to walk yourself through photosynthesis. On Blackboard, in the Our Classroom tab, there is a folder. I posted an announcement about this last week with supplemental study resources. There are videos in there about photosynthesis walking you through those different processes. Okay? It's all about making ATP. We were talking about catabolism. Once we've generated the ATP, it's all about anabolism and making other compounds. A cell is going to do and make whatever it needs in any part of the process. What you're looking at here is the end of glycolysis transition step TCA cycle. If a cell says, hey, wait a minute, I need the amino acid alanine, it can actually divert some of the pyruvate generated here over into making that amino acid. If it needs the amino acid glutamine, it can take a small amount of it from here and divert it over into making glutamine, okay? So when we talked about the maximum amount of ATP produced, that's theoretical. That's if the organism just starts and goes all the way through, start to finish, via respiration, or whatever it is. Most cells are going to go, wait a minute, I need to take a little bit off of that now. I need to do this. Oh, I need to take some of it off. Let me get some of that out. You ever went to the bank and deposited a check? And you're like, yeah, I want that money to go in, but I need you to give me like $40 back, okay? So you should have deposited this $100 check, but in the end, you're only actually turning in $60 because you want to keep some back. You make that decision what to do with your money, okay? Cells do the same exact thing. I know I could take this and I know I can convert it and I could make more ATP in the end. But I have to have this product over here. I have to have that product over here. So they will convert some of their compounds into making those other products. It's all a give and take. How are we going to study this? Okay? First of all, remember I have office hours. I am here Mondays and Wednesdays before your class. If you let me know you're coming, I will make sure to be on the lookout for you, okay? And let you know if I'm, get stuck in traffic, okay? Wednesdays I have office hours after this class too. Come and see me, send me your questions. Do not wait another week and go, wait a minute, I know we've covered 3 other chapters, but I'm still confused on metabolism, okay? Remember, metabolism is simply referring to all of the chemical reactions in the cell. It's not a specific term in any way. Every chemical reaction requires the use of an enzyme to occur fast enough. Everything that we have talked about in terms of what a cell would do, aerobic respiration, anaerobic respiration, fermentation, All of this comes back to the enzymes the organism has. If it's missing the enzymes in that pathway, it cannot complete that pathway. Okay? So the chemical reactions are controlled by the enzymes. The enzymes are controlled by things like temperature and pH and salt concentration. All cells are going to need ATP to build macromolecules. So if they are going to do biosynthesis, they've got to have an energy source to start with. They've got to be able to find a way to make ATP to then do that biosynthesis. How they generate ATP, how much they make, all depends on what they use. What do we think? There are some videos at the end here. In the interest of time, I'm not going to show them. Most of these are, I think, the first one for sure is the one from your textbook, okay? The biggest ways that I can suggest to study this are to try drawing it out. So last class we went from that side of the board all the way to this side. We said, okay, we're starting with glucose. What's our starting material? What are our end products? How does each one of these go? And we said, what's the purpose of each step? If you can do that on your own and explain it to someone else, you really understand it. So if you got, a group text, group message that you're working on, okay? You can say, I'm going to go first. We're starting with glycolysis. Glucose begins. Who wants to take the next one? You could trade off who says what and tag somebody, and, you need to explain the next step. Try explaining it to someone else. I cannot stress that enough. It makes a huge difference. Try quizzing yourself. Do the end of chapter materials. I do not suggest just reading the book because it is complex and it's going to be like, what am I reading? I don't understand. Okay? Watch a video. There's a million videos. Most of them are focused on eukaryotes. Remember, the difference is where they're happening. Prokaryotes don't have mitochondria. So glycolysis, transition step, TCA cycle happens in the cytoplasm, and the electron transport chain happens across the membrane. If you can distinguish those, you're going to be good to go. There are some of these, there must be some college class that requires students to make a song against respiration, and you can find all kinds. There's one that's to like this song, like Hey There Delilah, okay? I just remember the song. There are quite a few to 80s rap videos, like Baby Got Back kind of thing, if you're old enough to understand that. If you have never seen that, you should enlighten yourself. But if singing a song helps you understand it and be able to work through it, go for it, okay? The biggest thing you can do is to try and bounce ideas off of someone else. Here's what I got. What do you think? This is what I came up with. What can you add? Or I'm lost on this. Can you walk me through it? Work together. Come and see me. It will all be okay. All right? I cannot tell you how many times my husband had to sit there and listen to me work through different processes and different steps and how it all worked. And he'd go, no, you got that all wrong. Okay? And it was all fine. All right? My husband has a microbiology degree. All right? My husband was my lab partner. Right? I know, it was scandalous. But it was like we would bounce ideas off of each other. And there were some things he would get, and there are other things I would get. But that was the one way we made it work to study. I cannot learn by reading the textbook, okay? You've got the outline. If you've not picked up an outline this semester, go to the outline and try and fill it out, okay? See how far you get. It will make a difference, I promise you.
Oh, Mom.
Oh, and one other slide. This last slide here is also from a different textbook, but it does kind of outline your outputs. So where your total ATP is coming from, how much NADH, it says FADH, please keep in mind it's FADH2, okay? But it walks you through, and you can see how where it multiplies by 2, you can kind of figure out, okay, why are they multiplied by 2? How is that happening, okay? Again, if you understand what we're starting with and what our end products are, it's going to go a long way.